Let's recap some fundamentals before we critically appraise any paper. Experimental studies are the ones where researchers introduce an intervention and study the effects. Observational, on the other hand, are used to answer a research question based purely on what the researcher observes as the effect occurs naturally. Experimental studies can be classified as clinical trials, field trials, and community trials. Clinical trial can be further subdivided into randomized control trial and non-randomized control trials, also called preference trials, and have a Zillian design, a comprehensive cohort design, or Van Wels design. It is equally important to understand different types of randomized control trials. Randomized control trials can be classified according to different aspects of intervention evaluated. They could be pragmatic trials, efficacy or effectiveness trials, phase 1, 2, 3, and 4 trials. It is also possible to classify RCTs according to the number of participants. RCTs are also categorized according to the level of blinding. A single-blinded randomized control trial, where usually patients are blinded. A double-blinded randomized control trial, usually patients and treating physicians are blinded. Triple blinding means blinding of patients, treating physicians, and study investigators. Quadruple blinded randomized control trials, where patients, treating physicians, study investigators, and statisticians are all blinded. RCTs are also grouped according to participants' exposure and response to the intervention. It includes parallel design, which is the most common design, crossover designs, factorial design. The focus of this video is going to be parallel design because this is the most common type of randomized control trial. So we start with a sample population, it goes through randomization, and after that it goes to either an intervention group or control group, and then we compare the two outcomes. The study sample should be representative of the target population. Method section should give inclusion and exclusion criteria. The type of sampling and its logical reasoning should be mentioned. Details of sample size and its calculation to determine a clinically detectable difference, if a difference truly exists, must be given. Second question relates to randomization. Randomization is the key or cornerstone of a randomized control trial. The random transfer of subjects to a placebo or a treatment group is the foundation for determining the marginal difference between the two groups. The reason is that apart from the intervention under study, the two groups should be as similar as possible with respect to their baseline characteristics, prognostic factors, as well as other research methods, all to be applied equally and fairly. Randomization equally disseminates any confounding variables between the two groups, though some difference may arise by chance. So when critically appraising the RCT, the details of randomization must be present. The type of randomization should be mentioned and discussed, whether it is a computer-generated set of random allocations, blocked randomization if the sample size is small to certify equal numbers in each group, or where there is a potential baseline confounding variable, randomization could be stratified to equally distribute them between the two groups. Third question relates to if both treatment group and placebo groups are treated equally, for example in terms of frequency of follow-up and other treatments or protocol. They should be alike except for the treatment under study. You need to check whether all research methods appertain equally between the two groups. Another important question is, how is blinding accomplished? Blinding at the phase of employing the intervention and quantifying the outcome is important if bias, whether intentional or otherwise, is to be evaded. A reviewer must see if blinding is attained by making the intervention and control appear similar in every respect. The patient and the researcher should ideally be blinded to the study task, that is double-blinded. Sometimes, however, blinding is not even possible for participants, but researchers can blind themselves. Blinding can also break down due to numerous reasons, but this can be systematically assessed and limitations should also be discussed. Fifth question is how analysis of results has occurred based on the initial randomization, regardless of what may have happened to the study participants. This is described as intention to treat analysis, which means once randomized, then always included in the analysis. Therefore, no one is unfairly excluded or dropped. The study participants can drop out due to several reasons, like loss to follow-up, or may not react well to the intervention due to non-acceptability reasons. Details of how this is dealt or how loss to follow data is handled should also be mentioned in the study. How large is the treatment effect and the confidence interval? You need to see the precision of the treatment effect. You can see confidence interval. If it's too narrow, too wide, again, authors must not exaggerate or claim any significance in their results. Applicability or generalizability of results outside the research environment, that is, the local population. Additional limitations like small sample size, full dependence on self-reported measures, absence of blinding, all should be addressed and discussed in the paper. Quality control is equally important. Will all procedures are standardized? Details of data collection and data management to ensure avoidance of significant error, missing data, or inaccuracies. Were all clinical outcomes well thought out? Were all important members included, for example, commissioners, patients, healthcare practitioners, and caregivers, etc.? Are the outcomes and advantages in the real world discussed? Are benefits of the study worth the harms and cost projected and discussed?